Ladies and gentlemen, my role here today is to give a tutorial on confocal microscopy, and I'm going to take it at a very elementary level. So those of you, and I can see plenty of you in the audience who know all about it already, can leave now. I think that probably what most of you will want to know is if there's anything in this confocal microscopy for you. And I think the answer to that question at the outset is that it ain't necessarily so, but you'd better look into it to see whether it possibly does have anything for you. Now, I'd like to, could I have the projector on, please? I'd like to begin by telling you what confocal microscopies can do for you. And as far as I'm concerned, the most important thing, and why we got involved with it, can you hear me, by the way? Yes, is that it enables you to look at large and unprepared specimens. And that large and unprepared specimen is a dentist, and he's using a confocal microscope to look inside the enamel of his own front teeth. This is the operational head of that said confocal microscope. It's the one I'm going to talk about most today, the tandem scanning microscope configuration, and a TV camera is relaying the image to a TV monitor hanging from the ceiling so that he can see what's happening inside his own tooth. So it's the most important aspect then is that it cuts specimen preparation to a minimum, or at least that's the theory of it. In fact, if you look at your own teeth in this way, and particularly at this time of day, what you will see is the bacteria that make the plaque on the surface of the tooth when you focus into that level, and when you focus down further inside the tooth, you will be able to see the structure of the enamel prisms, where these little guys are about 5 microns across this repeating structure and represent what the cells were doing that formed that tissue at the time that they made it. And you can as well look at fossil material in blocks of rock as you can live teeth in live individuals. As another example of looking at a very large specimen, here I'm using a confocal microscope to look inside the bone structure of an elephant skull, and the entire elephant skull is on the microscope. And when we do that, we can look deep down inside the bone, and we can see the spaces inside the bone structure which were occupied by the cells in the bone. The osteocytes, the distance from there to there is about 20 microns, and these fine side-branching processes of the bone cells are about half a micron in diameter. We're focused well down inside the bone there in a reflected light configuration. Focus down another 4 microns and we see a different part of the canalicular branching mechanism of the first lacuna, and we see other lacunae coming into the field. And focus down another 4 microns, the first lacuna has disappeared altogether, and we're looking at portions of others. And since this can be done in real time, as fast as you can focus the microscope up and down, you can get a very strong three-dimensional impression from a microscope which gives you focal plane specific information. If you try to perform this trick with any other kind of optical microscope, you will see absolutely nothing. And that's the most important point about this microscope, that it enables you to pull out information due to very low intensity reflections or to fluorescent effects from inside bulk objects. Now, I'm afraid that these lights which are shone on me to make me appear on the video camera, and I think the video camera can well dispense without me, are spoiling the projection of these and all other fluorescent slides which I shall show you. But this is to show you a fluorescence image in a bulk sample looked at in the tandem scanning microscope configuration, and it's a piece of bone which was taken by a pathologist to look at the bone structure from a patient with very severe bone disease. The patient was given tetracycline antibiotic, which fluorescently labels bone growth fronts, and he was given at weekly intervals, so we have bone growth site 1, 2, 3, 4, 5 in sequence, and we're looking at a thin focal plane of fluorescence inside a big block of tissue. We focus down 25 microns, and we're looking at the same 5 lines, but we have a big hole inside the bone structure where the bone has been eaten away by bone-eating cells, and you will see from the selection of pictures which I show you today that that's a large part of the interest of our lab. Another 25 microns down and that hole is much larger, and another 25 microns down and it's much larger still. And I don't know whether the projectionist is going to take the cue of switching off the lights on me, but it really is going to spoil the pictures if we have so much bright light. Can you not switch off the floodlight approaching from at least one side of the room in order that we may see the slides? Now, there are many different configurations of confocal microscopes, thank you very much, which are now on the market, and they all do essentially the same thing. They enable you to look at reflection or fluorescence inside bulk objects. The latest ones which have come on the scene are specifically aimed at exploiting really the fluorescence imaging prospect and not even worrying about whether the sample is the bulk object, but using conventionally prepared light microscopic specimens like single cells or tissue culture cells, and being able to make much sharper images in fluorescence due to the fact that one can make an image of a thin plane within the specimen. And even within the range of thinnesses of conventionally prepared specimens, there's still a very strong advantage to using confocal microscopy, and particularly in fluorescence. And this is an example of a picture prepared using the Biorad laser sharp MRC, their latest configuration of microscope, of a specimen which is a section through retina stained with two different immunological stains for two different calcium binding proteins. And although this is a fairly low resolution picture, you'll have to agree that this is a very nice sharp picture that gives you a large amount of information content, and it's very much better than you could do with the use of conventional fluorescence in the microscope. Now, another thing that I want to say at the outset about this kind of microscopy is that the aim is to be able to produce information from very thin focal planes. But the ability to do that, quite surprisingly and paradoxically, also gives you the ability to make three-dimensional reconstructions, in fact to extract three-dimensional information from bulk light microscopic specimens for the first time. And here is an example of a crude and primitive way of making such a three-dimensional reconstruction. This is again a set of images made in fluorescence using the tandem scanning light microscope. And what I did here was to take 18 pictures on color transparency film material at 10 micron focal intervals of a specimen of bone which was labeled with tetracycline. You cannot see the detail here in this photograph, but there is exquisite detail in the original. And then I printed those transparencies onto 3¼ inch lantern slide plates and stacked them to make a three-dimensional reconstruction, which you can now hold and look through in any direction to see the complete three-dimensional structure. Well, of course, there are many other ways of doing that involving computer image processing and modeling, but that's just to give you an idea of the flavor of the thing. And in case I get very rushed at the end, as I'm sure I will, I want to tell you that not only is this the only way to use a confocal microscope to make a three-dimensional reconstruction which exploits and indeed even exceeds the resolution that's available in the conventional optical microscope, but these reconstructions have a very interesting projection property. They are parallel projections and not perspective projections, and are therefore very, very much easier to analyze geometrically. Now, confocal microscopy goes back in the literature, as far as we know at the moment, to this reference by Marvin Minsky, now of artificial intelligence fame, who filed this patent in 1957. So we're looking at him having already invented it more than 31 years ago, and it's taken really up until the present for it to become exciting for the interests of manufacturers. And I think there are probably now a dozen or 15 different confocal microscope manufacturers on the market, and some of them will be represented here at the equipment exhibition. And I think this figure 1 of Minsky's patent is probably the clearest diagram of the fundamental principles of how a confocal microscope works that's ever been invented, so let's use it to explain how it goes to you. We have a light source over here illuminating an aperture which is imaged by a lens into the plane which we wish to examine inside the specimen. Strictly speaking, onto the three-dimensional point or volume which we wish to obtain information from. We examine that uniquely and brightly illuminated point with a second microscope which is focused onto the same point. And the image of that brightly illuminated point is projected onto an aperture in front of a detector, here shown as a photodiode. And now one can collect information then from that point only, because if the light interacts with the specimen in front of or behind the plane in which we're interested, first of all, it's not very brightly illuminated, and secondly, the point which we're looking at will not be focused properly onto this aperture. The light will be well spread out, and so information from out-of-focus planes is strongly discriminated against in this arrangement, which today consensus has agreed to call the confocal arrangement. It's been called other things at other times. Now, one's only looking at one point in space, so it's necessary to scan this point through the object in order to be able to build up an image. And the way in which Minsky did it was to move the specimen past this fixed point in space by scanning it, attaching it to a vibrating mechanism driven by a loudspeaker coil to vibrate it in one direction, and driving it with a screw mechanism to get the frame direction. And with this kind of device, he could write an image on a cathode ray tube. He used a Navy surplus radar scope at that time. He could build up an image at about one frame every 10 seconds. And the many other microscopes which have been built on the mechanical movement principle have achieved frame speeds of about that value. And I'm afraid to say that, of course, that's the reason why they haven't taken on in many fields, because it is, in fact, painfully long to wait for one frame in every 10 seconds when the one frame only represents one of the many focal planes that you want to look at. You can accept one frame every 10 seconds in a scanning electron microscope because everything's in focus. But in a confocal optical microscope, you've got to go through all the many planes in order to find the information that you want. So 10 seconds frame speed is hopelessly slow. He also indicates here a reflected light configuration of the same thing. Here's the illumination pinhole, a beam splitter, and the returning light put onto a pinhole in front of the detector system. Well, now, Minsky got that going. He demonstrated it to many people, but he never published it in the proper scientific literature, and he never even bothered to record a picture that was taken with his microscope. Many years later, Shepherd and Wilson in Oxford and Brackenhoff and his colleagues in Amsterdam in particular, together with others, took up this work and improved it a great deal. Here is a picture taken from Shepherd and Wilson's initial attempt to go commercial, showing how you can scan the specimen past a stationary optical spot, put the returning light into a detector confocal pinhole in a reflection mode. The thing began to be much more interesting when people improved the frame speed by scanning the light beam rather than scanning the specimen, using some configuration of mirrors. And this is a picture taken with Zeiss's first confocal configuration, which I took on exhibition in London a couple of years ago, showing you a portion of the branching dendritic tree of a Purkinje cell in a confocal image. That's taken with a mirror scanning microscope, where they could crank the frame speed up to about 1 frame per second maximum, which is enough to scan through the specimen and find out where you are. Today there are many manufacturers of mirror scanning microscopes that are operating at different speeds, and Heidelberg Instruments is just yet another one of them. That's the Phoibos instrument coming from Sweden, for example. But all of these microscopes which operate by scanning mirrors are really only able to crank the frame speed up to a second or a fraction of a second if they go to a reduced pixel resolution. But coming down the line, we can see some real improvements when the optical beam deflection is done with an acousto-optical deflector. And this is a diagram taken from the work of Haupt and Dreyer in Delft in Holland, who have built a confocal microscope using an acousto-optical deflector on one axis, and they are able to, for the first time with a laser scanning microscope configuration, utilizing nicely the very high intensity of illumination that can be obtained with a laser, they are able to produce images at real TV frame rates. And that's a very important step forward in the use of lasers as the illumination. And these are some images which I recorded on their microscope on a visit there some months ago. And it's labeled what they are underneath. Where I've put that the image is a range image, it indicates that we've not only taken the one focal level, but during the time that we record the image, we've actually changed the focus to add together the information from several different focal levels. And that, as I shall hope to remember to explain later, is the way by which you can extend the focus range of this kind of microscope, yet maintain the full resolution and clarity, because you are only adding together a series of images of image planes which are really perfectly in focus. And just to indicate how discrete this information can be, these are two images which I took back to London and processed on my own Tracor image analyzing computer of one focal level in a specimen recorded in autofluorescence in 2 seconds total integration time, and the same field of view with the 2-micron focal level difference. You see a completely different image in this area that I was pointing at. That's exactly the same area of the specimen. Now, the very highest frame speeds that have been obtained in confocal microscopy are those obtained with the tandem scanning reflected light microscope of Petran and Hadravsky of Pilsen in Czechoslovakia. And in case you don't know it, Pilsen is as famous as a brewery town as Milwaukee, Wisconsin is in this side of the pond. Now, it was in Pilsen, Czechoslovakia, that the confocal microscope was invented for the second time, but with a totally different configuration. This shows their microscope photographed 15 years ago already in the middle of a physiological experiment. And they invented their microscope already 21 or 22 years ago in order to be able to look inside bulk living tissue. They were not concerned with looking at fixed and prepared specimens, as Minsky was. Minsky tells me that he invented his microscope to look at Golgi preparations of the brain, and he was interested in trying to find out how the brain worked by having a better idea of what the three-dimensional wiring diagram was. And it was exactly the same sort of idea which led Petran and Hadravsky to invent their kind of microscope. Now, since that time, of course, on beam have come Tracor Northern in this country with the first microscope manufactured outside Czechoslovakia, who exhibited here last year and have some microscopes on the exhibition here. But this picture is taken of a portion of the disc from the Pilsen, Petran-Hadravsky microscope, and it's a photograph of the aperture array which is used to illuminate the specimen. Now, in this microscope, the scanning is done by scanning physical apertures which are placed in the intermediate image plane of the objective lens. And these apertures that you see here, there are many tens of thousands of them in an extremely thin disc, 4 inches in diameter, of which you see the periphery here. And the band which contains these holes is roughly 2 centimeters in diameter. Each one of these holes is unique in the sense that it lies at a unique radial distance from the center of the disc, which will be clearly up there several yards into the ceiling. So that as the disc spins, each one of these apertures being illuminated scans a single line in the focused-on plane in the specimen. These apertures, being in the intermediate image plane of the objective, are imaged in the focused-on plane at the specimen at a reduced magnification equal to the magnification of the objective. So if we have a 100 times objective, and each of these apertures are, say, 30 microns in diameter, then they will be imaged at 0.3 microns in diameter in the focused on plane in the specimen. A similar array of apertures are used to detect the returning light from each of the illuminated points corresponding to each one of these apertures. And the way that that's done in the Petran microscope is to use the opposite side of the disk and to exploit the fact that the disk is so constructed that for every one hole that has a particular radial distance on this side of the disk, there is on the same diameter on the opposite side of the disk a hole at the identical radial distance, and that identical hole is used as the detector aperture. So there's a one-for-one correspondence between all the holes on this side of the disk and all the holes on the other side of the disk. Now, this device clearly has some central symmetry, so the other side of the disk is back to front, top to bottom from this side of the disk, so that inside the microscope there must be a mirror assembly to create those inversions in order to overlap the two sides of the disk. And the way that it's done in the Petran-Hadravsky microscope is illustrated in this diagram. The disk is this feature here labeled N after Nipkow, and the illuminating light comes in here off a steering mirror through a field lens just above the disk. The light then passes off one mirror, off another mirror, through a beam splitter, off another steering mirror down to the objective. And the only purpose of this particular mirror here is to make the objective point downwards, since the essential concept of this microscope is that one will be using immersion lenses to look at live specimens, and so they're going to be water immersion lenses, and water runs under gravity, so the objective must be upright. Otherwise it could stick out in this direction here. The returning light comes back through off this mirror, bounced off the beam splitter here, off a final mirror, and is imaged onto the disk on the opposite side, where the disk is examined by a Ramsden type eyepiece, that is an eyepiece which focuses in front of itself, looking at the plane of the disk. And now, just as in Minsky's diagram, we have a one-to-one correspondence between illuminating aperture and detection aperture, but in this case we have 1,000 points or so illuminated and detected simultaneously, so essentially the whole device starts off by being 1,000 times faster. And that actual frame speed is determined by the disk rotation speed. The disks can be cranked up to speeds we don't know what the limits are yet, but it's certainly possible at the moment to have frame speeds of several hundred frames per second, and that can almost certainly be put up to several thousand frames per second. And I think there are two interesting things here that I'd like to mention to you as a diversion. Petran and Hadravsky attribute the design of their aperture disk to Nipkow, who took out his patent in 1884. That's 104 years ago now. In fact, they're very generous in assigning it to Nipkow because their development of the Nipkow disk is an extreme development of the original idea. But I'd like to point out to you that this patent of Nipkow's in 1884 was a complete patent for television, a completely functional system for television. And that patent was translated, I believe, almost word for word by James Alexander Logie Baird, the putative inventor of television this century. And all he did was really to copy Nipkow's patent and assume that nobody read the patent literature. And that was 40 years after, and now we're 30 years after Minsky's invention. So you can see how you can lose anything by putting it in the patent literature. Now, why didn't the Petran-Hadravsky invention catch on at the time? It was certainly because the Russians invaded Czechoslovakia in 1968, and that meant nobody could visit them and they could visit nobody else. So they went on with their development of the microscope in total obscurity. But it was also because in their initial paper describing the results of this microscope, published in Science in 1967, 21 years ago, they only illustrated it with drawings because they found it hard to convey the excitement of being able to get information from individual focal planes and put all this information back together. So what they did was to make composite drawings from focusing down through their live specimen. Years we, recorded pictures with photographs this century, not with drawings, and that may be one of the reasons why it did. Now, there's another way in which you can use one of these Nipkow discs, and that is to use only one side of the disc for illumination and detection. And that's a pretty obvious thing to do, and it's obviously a much easier thing to line up than all those mirrors in the Petran-Hadravsky configuration. And the disadvantage of this arrangement is that you have 1% transmission, say, in a typical one of these discs. 99% of the light is stopped by the solid part of the disc. That means 99% of the light is reflected back off the top surface of this disc and has got to be discriminated against the light that comes from the sample. Well, you can do that reasonably well using a polarizer, a quarter wave plate, and another polarizer over here to discriminate against the light reflected from the top surface of the disc, which didn't pass through the quarter wave plate. But never mind, in fact, the level of reflections which you get from biological samples are so low that this system of discrimination is not useful in the biological world. But it is if you look at a very high intensity reflector, like a piece of silicon. And if you're interested in using one of these microscopes to image the reflection from a surface, which, as I shall show you shortly, is also a very interesting thing to do, then this is a useful configuration. They can also stop a large part of the unwanted light because it's brought to a focus at a different point than the light returning from the specimen by putting an opaque stop in the center of the device, and they can also tilt this disc with respect to the illuminating beam. But never mind, they haven't yet shown that it will work on biological samples. Now, I believe that the tandem scanning reflected light microscope configuration is a very useful one because it gives a real-time image, and I want to give you an example here of just how fast this frame speed can be. This is my colleague Tim Watson, who's a dentist, who's using the microscope here to watch teeth being drilled by a very high-speed drill. These drills rotate at 500,000 revs per minute, and he's configured a stage here where he can look at the end of a dentist's drill, watch it moving into a tooth, and watch the damage that's happening inside the tooth structure ahead of the point that it's happening. This is recorded on a videotape and analyzed on slow playback. So this is what happens in a still. You see the enamel structure here, you see the blade here, and what he's looking at is what's happening in this superficial zone as the destruction is occurring in real time. I believe that it's a very long way from the possibility to do that with a single beam confocal microscope. You need to have a multi-beam approach to get the frame speed up. I want to emphasize that what you can do, apart from the frame speed aspect, what you can do with one kind of confocal microscope, you can do with another. And this is to show an application of confocal fluorescence in the tandem scanning microscope, where we're looking at the junction between a dental filling material down here and tooth dentine and tooth enamel here, where the bonding agent that's used to bond the filling to the tooth had a yellow fluorescent agent added to it. And in this case, you can see how the fluorescent agent has penetrated the surface of the dentine, the surface of the etched enamel, and into the plastic of the filling material over here. I'm only using this as an example to show you the difference between confocal fluorescence and ordinary fluorescence. The next picture shows exactly the same field of view of exactly the same specimen, the same lens, without confocal imaging, and you cannot discriminate any detail at all in an ordinary fluorescence image, epifluorescence image. There's another such an image of the bonding of a fluorescent agent to dentine. These tiny canals in dentine are about 1 micron in diameter. And here we're using blue light to illuminate the specimen, and we're looking at the blue reflected light as well as the yellow fluorescent light. Here we've tagged the bonding agent with rhodamine, and we're using green irradiating light, but we've got the appropriate red filter in the output, so we only see the fluorescent light. All these tricks are possible. We can combine reflection and fluorescence images simultaneously, wanting to do so. This is the image of a bone implant made of titanium. This is a titanium screw, against which bone has grown. This tissue is bone. We have a very high intensity of reflection from the metallic titanium and a low intensity of fluorescence from the fluorescent dye used to stain the bone and the bone cells. But we can image both together by a suitable discrimination with the pass filter against the illuminating blue-violet light. Now, the error has been propagated in the recent literature in spite of my personal energetic protests that this is an error, that in fact the confocal tandem scanning kind of microscope cannot do fluorescent imaging. This is a profound untruth, and I'm going to give you an example here of how comparable the confocal scanning laser microscopes are with the tandem scanning microscopes. Here's a set of confocal images taken at, I think it was half-micron focal intervals through an osteoclast. It's been labelled with FITC labelled antitubulin, I think it was, and we go here, you'll see that this cell has a very large number of nuclei. Those are the cells in which we're interested in. That was taken in the Biorad laser sharp MRC microscope. Here is an image taken in an ordinary fluorescence microscope of a rhodamine labelled cell. This is non-specific rhodamine labelling, I'm afraid. An osteoclast, you can see that it has red labelled nuclei. And that's just an ordinary fluorescence image. It would look better with the room lights out, but there we are. There's a set of confocal fluorescence images recorded in the tandem scanning microscope at 1 micron focal intervals in this particular case. And you can see how different these images are from one focal plane to the next. These are recorded with a very sensitive camera looking at the fluorescence image. The fluorescence intensity in this case was so very low that we couldn't even see the fluorescence with the naked eye, but with the appropriate cooled CCD camera, we were able to record these images. And I believe that that camera may be on display at the exhibition today. And although there's been a failure in the photography here, there's one image of that set color-coded in order to obscure the real information content, which is, of course, the main purpose of using color coding. There is a fluorescence image of FITC labeled antitubulin in a bone cell. This is not an osteoclast. There is a set of through-focal images. I think it's the same cell. It's an awfully similar one, if not, recorded at 1 micron focus intervals using the tandem scanning microscope with the cooled CCD camera, and the images show these bundles quite clearly. There are a couple of images of that set, color-coded again, in order to, as I say, prevent you seeing what's really there. Now, I'm afraid we're really wiped out by the light on that one. If I could beg the organisers here to switch the lights out, we can actually read this slide for a moment. This image is not a fluorescence image, but a reflection image recorded in the... it would be nice if you could switch me off and switch the slide on for a moment so that people can read this, because there's a lot of very fine detail in this picture. This is a reflection image of a immunogold labelled cytoskeletal preparation of some cells. I've forgotten what cells these are. And this is an original 70mm format negative recorded directly with the light coming from the tandem scanning microscope. And you can see that we have very nice detail here. This is from 1 focal level inside these cells. So that kind of resolution is also possible with all the confocal microscope configurations. And if we can keep the light down just for this one, please, I'd be most grateful. Now, another characteristic of the confocal microscopes, which didn't interest me to begin with when I first started to look into using them myself, is the very sharp contrast that you get at focus and the effect of reversal of contrast as you go through focus. Now, in order to demonstrate that this is true and over what range it's true in the tandem scanning microscope, here I've illuminated our Tracor microscope, which we acquired late last year, with an old sodium vapor microscope lamp. It's a very low intensity lamp, and I haven't got the photographic exposures right here, but these are direct photographic exposures of a 10 micron checkerboard pattern. This is a calibration standard for SEM in silicon, and I'm looking at the same field of view with a half micron change in focus, and we can get such a very precise change in focus from the piezoelectric controlled focus device on the objective of this microscope. And we could get this change in contrast, the white checkers being black in this picture here, with just a half micron shift in focus. Now that very dramatic effect is demonstrated by all the confocal microscopes, a reversal of contrast as you go through focus, and a very sharp contrast. But there's a very interesting effect which is, I think, presently unique to the tandem scanning microscopes. Both the one-sided and the two-sided configuration show this effect. And that stems from the following. If we go back to this diagram which shows how the microscope works, we illuminate a single aperture and image that with a lens into the focused on plane of the specimen. If we start with white light, we have to contend with the longitudinal chromatic aberration of the objective. So the white light is split so that the best confocal position for red light will be here, for green light will be here, and for blue light will be here. So that if we move a reflector in this direction with respect to the objective, the confocal condition will be satisfied most perfectly for blue light here, for green light here, for red light here. In other words, the reflector will change color as we go through focus. And so we can use this effect to produce a direct color coding for depth effect. Now this set of images are recorded illuminating the Pilsen TSM with a mercury arc lamp. And in the mercury arc lamp there are very strong lines, and the color is dominated by the color of those strong lines in the mercury arc. We're looking at a bit of silicon chip here, and the focus difference between this plane and this plane is 1 micron, 1 micron, and 1 micron. And notice the dramatic color changes of corresponding strips here as we go through focus. But notice also that all parts of the surface which have the same height at any one focus level are color-coded in the same color. And just to make sure that this color was due to the reflection and not to the natural color of the substrate, I had coated the specimen very heavily with metal in order to remove any natural color effects. Now, in fact, that turns out to be quite interesting for some of the work which we do in my laboratory, and we're mostly interested in the last few years in the processes of how bone is resorbed. And the kind of experimental system that we have is to take isolated osteoclasts, bone-eating cells, and put them down onto slices of bone or tooth. This is a rabbit osteoclast which we put down on a slice of sperm whale dentine 24 hours ago, where it had its breakfast and then its lunch, tea, supper, midnight snack, and it's having breakfast 24 hours later. And we want to know how much of the dentine or bone these cells eat. And the procedure that we've been using for many years now is to take stereo pair scanning electron micrographs with high tilt angle difference. We've constructed a special stereo photogrammetry equipment to get high-speed measurement out of these things. And so we can, by essentially air survey photogrammetry, measure up the volume of these pits. I found this picture in a magazine recently. It shows a swimmer against a sperm whale with some teeth sticking up there, and I thought that was an appropriate diversion at that point. Now, it turns out that we can look at those similar specimens in the tandem scanning microscope and have information about the relative depth of the surface simply from the color of the reflected light. So here's an image where we're focused at the surface of a slice of dentine with a hole eaten into it by one of these cells. And you'll notice the color difference in the center of this pit. We focus down 3 microns, and we have a dark surround and a different color for the different parts which are in focus. If we look at this as a black and white image, the part which is in focus appears white, and the parts which are out of focus appear very dark. So we can use this color coding for depth then to measure things. But we don't believe that's actually the way to go, because we now have a much more precise way of doing that. And I'm going to demonstrate that to you with an example here, which we first measured using scanning electron microscopy. First we make a video film of the interaction. Here we've picked up an interaction where we've seen an osteoclast which has eaten 3 holes, and we have the time sequence here. I think we follow this over 14 hours, and during those 14 hours it eats away this amount of dentine, and we can follow that in the videotape. So there's the cell which did the eating, and then we convert those into numbers. We can find out how many cubic microns per minute of bone that particular cell can nosh away at over different time intervals. We can find out how long it takes to eat its own volume of tissue, for example. And I've taken that same sample and produced this color-coded contour map using the Trakel Northern TSM coupled to their Trakel Northern TN 8500 computer. And for this I'm deeply indebted to the people from Trakel Northern, Chuck Dillon, who's out there in the audience, in particular for developing the software to make this possible. Now what we've done here to measure the depth is to exploit the fact that in any confocal microscope, you could do this with any of the laser scanning microscopes too, with the appropriate software, you have a maximum intensity of signal at focus. So that if you go through a whole set of focal planes and ask the question at every pixel, is it now brighter than it was before, then reset a frame store image value to that new value, then ask the question again. You can go through all the values and find the focal plane at which the signal intensity was brightest, and set a value in a height image equal to that particular focal plane increment. So here we have a map which shows a total range of 12 microns, from white through grey, these mauve colours, down to black at that point. And we can read out the height value at the position of the cursor. So where that cursor is, we read that the depth with respect to the top position that was found is 11.62 microns down. And this focal plane readout can be done with a nominal precision of 4,096 parts in 50 microns, certainly anyway very much better than 1/10 of a micron, and now represents an entirely automated procedure for mapping surfaces and also for determining the volume of the feature, since the volume of the feature is the height of all the pixels, as it were, below a reference plane aimed at the edge of the pit feature. And you can play with those images to your heart's content. There is putting contour lines at 2/3 of a micron intervals through that image. You can, once you've got the information set, reproject it from any other angle of view. You can project it from a large number of different angles of view and generate a pseudo hologram. And those pseudo holograms, some of them I believe are on display on the Trakel Northern stand, where you can go and have a look at them. You'll need to stand a bit closer to them than you are given an opportunity there. This is one which was done, one of the earliest good examples which we have. It shows a very large resorption pit in a surface of sperm whale dentine. We've color-coded the different slices here. We can play around with the display mode there. There it's zoomed up, and we can read the height at a particular pixel in the bottom of the image there. You can change the colors. You can have it in black and white if you're colorblind. You can have that kind of display. That's the kind of thing you can do when you've got that ability. And I think this is going to represent one of the major ways in which confocal light microscopy will go, that you've got this unique ability to get exact three-dimensional characterization of surfaces, be they real surfaces or internal surfaces. I'd like now to examine the possibilities for cross-correlating confocal microscopy with other microscopies which we want to use, and I think we can do this in any direction. I'm going to illustrate here cross-correlating backscattered electron imaging. This is a picture of a polished, embedded piece of bone. And bone tissue, in case you don't know it, isn't with you for life. You keep turning it over. Half-life about 15 years in an adult. And the bone which has been with you for longest is densest and shows up whitest in a backscattered image. So that's old bone, and this darker bone here is bone which has been formed at a later period. Now, that montage field is identical with this montage field taken in the TSM. Here, this preparation we stained with a stable fluorescent dye, and we're looking at the fluorescence image of brilliant sulfur-flavin is the name of this dye, which we started to work with because it's also cathodoluminescent. You can make cathodoluminescence images showing this distribution. But here we're able to make this fluorescence image in the TSM even at depths deep to the surface of the specimen. And in fact, if you've got a TSM, this is a very much easier way to study the variations in bone density, because it turns out that the areas which are brightly stained with this dye are the less dense areas of this bone. And we have that exact correlation with scanning electron microscopy imaging. But more exciting in the recent past for us has been the development of methods for cross-correlation with other methods of light microscopy. Now, again, the more recently produced confocal scanning laser microscopes designed for biological users make it very convenient to find a cell using conventional optical microscopy, for example, dark field or phase contrast illumination, and having found that cell, to capture a confocal, usually fluorescence image of some immunolabel that you've added of that same cell. And I think, to be slightly acid at this point, it's a jolly good thing that they can find the cells using some other method of illumination, because if they were to find them with the comparatively slow frame speeds which are available, and with the considerable damage which can be done to the fluorophore with the laser beam, then they wouldn't be able to produce the very fine results, which in fact they can produce because they can find the cell with some other means in the first instance. Well, those people have been putting it about that this, which is easily possible in what I would call the black box configuration of laser scanning microscope, in which the scanning beam is introduced from a separate box into the phototube of an existing conventional microscope, and you take the return signal out and descan it in the same black box. Those people have tried to represent that that's a unique advantage of their system. It isn't, of course, and we can also do that sort of thing in various different ways in a tandem scanning microscope. Let me show you two different ways. First of all, we can use the tandem scanning microscope to illuminate another microscope. Now here we have a dish with some sea urchin eggs in it, and the problem was to locate those sea urchin eggs. And it's very difficult in a confocal microscope to find something in three-dimensional space because you don't see it until it's in focus. So we put an inverted optical microscope underneath there, and then when it was the water immersion lens here was brought down into the dish, we could look through the inverted optical microscope and see the image using the TSM as a non-confocal source of illumination for this microscope. And using that configuration, not the same specimen, here is a specimen of muscle stained with silver cholinesterase to demonstrate these muscle end plates, using the tandem scanning microscope to illuminate the specimen in transmission. This is the non-confocal image, and we could see at the same time, looking through the TSM, this reflection image from the same preparation. So we could look at both sides of the specimen at once in the TSM. Here's another way of getting this cross-correlation, and that is to use an additional source of illumination for the tandem scanning microscope. And this shows an early attempt to do this with the Tracor microscope, an old Watson light microscope lamp and Watson substage condenser bolted onto the microscope, so that we can illuminate here and use virtually any standard method of transmitted light microscopy that you know of to look at the sample in non-confocal mode at the same time as looking at it in confocal fluorescence or reflection mode. And let me give you some examples of what we can do with this and how this cross-correlative method can be very useful. Here's a picture of hamster cortex, a thick section. The section was 250 microns thick, and we're looking here at some blood vessels, and we see portions of the same blood vessels which are much more than 100 microns away in the Z direction. The same field of view in confocal reflection, we only see those portions of the blood vessels which lie in the thin focused on plane. If we have both methods of illumination running simultaneously, we can see the confocal reflection image in the context of the transmitted light image. Here is a picture of a ground section of a tooth. That's a sawn section, perhaps 150 microns thick. These very thin lines here are the dentine tubules. The finest branches that you see here are down to less than 1/3 of a micron in diameter, and normally extremely difficult to resolve in optical microscopy. They're very easily resolved in a confocal microscope. Over here we have the enamel prisms seen sideways on. We see the reflection from the enamel prism boundaries. The same field of view in the same microscope at the same time, and remember these are real-time images in real color in the TSM, seen between crossed polars. And we see a unique extinction direction in this inner layer of the enamel here, for example, which is why it appears very bright. And then we can put the two images together and color code one. We've color coded the polarized light image in red here so that we can see the polarized light information and the transmitted light information simultaneously. To show you that this is not only of interest in biology, this kind of microscopy, but across a wide range of applications, here's a geological sample. And we're looking at this geological sample in reflected light, focused a little way into the top surface of a rock section. The material was igneous dolerite. And I want you to pay attention to this crystal grain here that I'm pointing to, and this dark crystal grain here. Now, on the same occasion, we see those two crystal grains there and there in ordinary transmitted light. Here we see those same two crystal grains in ordinary transmitted polarized light. And remember, we can mix these signals in any proportion. Here we see the transmitted polarized light mixed with the reflected light image from the TSM. But another thing we can do in the TSM, which has not been demonstrated to be possible in any of the laser scanning microscopes so far, is to do reflected polarized light. And if you can remember back to the first image, here we can see this grain very clearly, which was difficult to discriminate in the first instance simply by using polarized light in the illumination system of the microscope. Now, if somebody could jump the second carousel on there, I'll proceed with this picture display. Right, another example of this cross-correlation. Here is a hematoxylin and eosin stained section of some tissue which has been labeled with, radio-labeled, with a compound which homes in on sites of low oxygen tension in tissues. And I think you can see the black dots of the silver label at the surface of the section here. Now, if you look at such an autoradiograph in the TSM, you get extremely bright reflection from the silver grains. And now I'm afraid I forgot to put the next slide in, you can turn up the intensity of the reflected light and you can read the hematoxylin and eosin stain, which comes from non-confocal illumination and so has a great depth of field, combined with the reflected confocal image of the autoradiographic grains at the surface of the section. They appear to be in focus at the same level. They're not, of course. We're exploiting the fact that the confocal image has a very shallow depth of field, and we can combine that at the same time with the large depth of field of the non-confocal illumination, which we can control by controlling the aperture of illumination. And I've calculated that we must be able, even at quite high magnifications, to get at least a 30-to-1 difference in the non-confocal depth of field compared with the confocal depth of field running simultaneously. But another advantage stems from being able to look at both sides of a specimen at once. Here's a pyramidal cell in rat cortex, looked at in ordinary transmission. You'll see fibers out of focus at many different planes. Here we have combined the confocal image, which appears orange, with the transmitted light image, which I've color-coded in green, and now we can see these projections from the top surface of this cell for the first time. We were never able to see them before in routine optical microscopy. Now I guess that signal is probably telling me to get a move on, but it's also telling me to ask the person who switched that light on to switch it off again, because the next view will be much better seen if we have the light down. Other recent things that we've found with this microscope are that it's very useful for looking at cells in the tissue culture situation. It was now quite a surprising thing to find that we initially conceived of it to be useful only for looking at bulk objects, but in fact it is very useful for looking at very thin objects too. And here is a tissue culture cell. It's been stained with an acid phosphatase reaction to demonstrate that it is an osteoclast, but whether or not the cell is alive or dead or has been stained with any other stain, we can see the distribution of very close contacts of the cell with the substrate in this kind of microscope. We can see these so-called focal contacts of cells which are migrating on substrates with very high clarity and very easily, and the instrumentation is a great deal less complicated than the reflection interference microscope methods which have been used to demonstrate that before. Here's another osteoclast seen in transmitted light, acid phosphatase reaction. There it is seen in confocal reflection, and you can see these interference fringes here, which probably relate to the thickness of this very thin peripheral portion, cytoplasm of the cell, which is spread out on the flat substrate. It doesn't matter what the substrate is. Here is an osteoclast which is well stuck in to eating a piece of dentine, looked at in transmitted light, and the same cell looked at in confocal reflection. We can now see the true distribution of the cell. We can see all these fine peripheral cell processes, and we can see these very thin singly nucleated cells on the substrate surface, which we were not at all able to detect in the transmitted light image. This is an osteoclast stained with toluidine blue, and the same cell looked at in confocal reflection. You now pick up these very black areas, which are where the cell is in very tight contact to the substrate, and a great deal of other information which is yet to be interpreted. This picture is especially for the stereologists, and Dominique Gelin is sitting in the audience. Once you've got one of these microscopes, you can do many other things as long as it's attached to a frame store. This is an image of bone in which we have subtracted an image taken at one focal level from an image taken at a focal level 4 microns away. If you do this, you show features which are present in one focal plane but not the other as white, and in the other focal plane but not the first as black. And if you want a proper stereological procedure for counting points in space, this is the basis of doing it, you should count either the black ones or the white ones. You don't see the ones which are present in both focal planes because they show as grey when you perform this technique. You can do any other image manipulation. Here is, in fact, an image taken with the old Czech microscope, which, as you've probably noticed, give rise to strong curved lines which are not present in the new Tracor microscope, with the curved lines, which have a particular spatial frequency, removed by making a two-dimensional fast Fourier transform, then putting a filter on that and a back transform, and we've removed the scanning lines. The same Fourier manipulations can be used to determine spatial frequencies, so we can measure the size of those features that we saw in that previous image. At the outset, I said that a very important thing would be three-dimensional imaging. In all the confocal microscopes, it's now easy to do this. It's particularly easy in the tandem scanning microscope. The basis of procedure is simply this. I've drawn here a biological specimen with a cover slip and a slide which has exaggerated depth. And say we want to image this three-dimensional volume inside the sample. All we need to do is to start recording one photograph with the stage focused here, and then keep on changing the focus while we record that photograph and stop the exposure there. Wind on, take another photograph starting at the same focal plane and focus down like that. Now, if you do this trick with a tandem scanning microscope, you've now recorded in full color, in reflection or fluorescence or any combination, a high-resolution stereoscopic pair. I haven't got stereo projection facilities here, but I'll show you some examples. This is a mechanical device with which we first made such stereos, cobbled together out of sawn-up old light microscopes. You can see two fine focus mechanisms bolted together at an angle. We now simulate that with computer-controlled motors. We can change the color of the illuminating light as we change the focus. And to demonstrate that this works, here's one in which we've simply changed the color through the rainbow sequence of red, orange, yellow, green, blue, in focusing down through a preparation of Purkinje cells in mouse cerebellum. So everything that has the same color is at the same depth, and we've color-coded for depth there, a range of 40 microns. There's the stereo pair partner. If we had stereo projection, you could see that. Another way you can do this is to take a single set of focal plane images and then to shift them relative to each other to reconstruct a stereo pair. This can be done in real time. This particular example done on the Tracor Northern instrument. The Biorad Polaron people have cottoned onto this idea and can reconstruct stereo images from sets of images which they have to store first in order to make this manipulation. You can also make stereo images with a device like the Tracor instrument through focusing using piezoelectric control of the objective, for which they now have extremely high precision. And here is one member of a stereo pair of a 10 micron thick slice in the middle of a 250 micron thick slice of brain. That's one member of the stereo pair. There's the other member of the stereo pair. If we had stereo projection, I'd show it to you. There's a stereo image of fluorescence image of bone cells. There's the other member, that's the fluorescent stereo pair. This is a stereo pair reconstructed at very low magnification. I want to emphasize that confocal microscopy is not only about high magnification applications. It can be extremely useful right down to the area where we could work with a macro camera, with a 35mm macro lens. And here's an example of trabecular bone. That's one member of a stereo pair taken by taking a confocal set of images and then reconstructing the stereo. There's the other. Now, in summary, confocal microscopy is interesting